All right. Oh, is that me? Did I forget to turn that off? Darn it. I think that is. That's what I had said, right? Was test next Thursday? Yeah. Yeah.
Oh, okay. I taught it in this week. I didn't know.
Yeah. So let's see. Modules, exams. Yeah, the 7th.
Oh, okay.
Yeah. Maybe because I hadn't turned it on. It may not be in the calendar. It might, or I don't know. Okay. Not a big deal. You know now.
You know I have to call code.
That's okay. We're going to try and do it in the classroom this time and not in the computer lab. So I really want you guys to bring your devices with you Thursday this week. We can do a little trial run of stuff again, whatever one's got the lockdown browser. And then the other thing is we are going to do an activity. You guys are going to get in groups and stuff like that, and so you might want to have access to the internet and whatnot. Anyways. So I think the problem for you, Annie, was not being able to get onto the Wi-Fi, but I said I just log you on to my account. Okay. And I can do that for anybody who's, like, if that's the issue, that I can fix. But the lockdown browser thing, I can't. So I don't know where we stand on that, but I want to get started on lecture. So I think last time we didn't get anywhere, huh? Really didn't get anywhere. Okay. All right. So maybe just kind of start at the beginning. So we all know about DNA to messenger RNA to protein, though. You guys all feel pretty comfortable about that flow of information, what we call central dogma? Anyone not feel comfortable about that? Okay. So the important thing there is we talked about this code, right, and what and how the code is read to make proteins. So we looked at this, I know we did, right? And we looked at how that code, those 3 nucleotides and messenger RNA, and how do you know that we're looking at RNA? Anyone know? The U's, right? U's are only in RNA. If we were looking at DNA, what would you see?
T's.
You would see T's instead. But you still see A, C, and G for both of them, right? Those nitrogenous base, those nucleotides. So we know the code, right? There's 64 different codons. Got your message, you're good. And so three of them are stop codons, which tells the ribosome to do just that, stop. One of them is a start codon, which is AUG. I just always remember AU. I don't know. AU guys. Like if you want to start a conversation, you usually have to hail someone first. I don't know. It works for me. Yes, everyone laughed at me at the workshop. I'm always like this. That was the comic relief sometimes. But there were other people that were quite funny too. So the rest of them all code for amino acids. So this code you see here, this 3-letter code, codes for one of the 20 naturally occurring amino acids. And so if we have any changes in this code, it could change which amino acid the ribosome puts there next. Right? And so that's where, you know, change can sometimes happen in the DNA and not actually change the protein that's produced. Other times, as we'll see, that code changes in the DNA, so therefore the RNA is different and the protein is different. And so this is really important. And then when we look at mutation, why sometimes even if we get change at the DNA level, you don't see it at the protein level. Still with me? Okay. So we'll look at some specific examples of that. So there are 3 different ways in which bacteria can adapt to changing environments. And what happens is, remember, they're constantly changing. And the environment also changes. So as the environment puts that pressure on it, it selects who's going to survive. And so the most important selective pressure, one of the main reasons for the workshop that I went to last week, is this whole problem with resistance to antibiotics, right? So how did they become resistant? Well, they genetically changed, which they do anyways. Why do we have so much resistance nowadays? Because we've used so much antibiotics and used it in ways that we really shouldn't have been using it, right? Feeding it to chickens even when they're not sick. Really not necessary. So then the microbes in the environment are overexposed to the antibiotics, so we are selecting, therefore, for the ones that are resistant to the antibiotics. That make sense to you guys? Right. That's one of the main reasons actually we teach about bacterial genetics, is this problem of antibiotic resistance. So we're going to look at specific mutations, right? Changes in the DNA, and how does that affect the organisms? So we're going to look specifically at base substitutions. And when we say base, we're talking about the nitrogenous base. We're talking about the A's, the G's, the T's, the C's. We're also going to talk about if you remove one of those nucleotides, or if you add an extra one in, what effect does that have on the code? And it creates a specific type of mutation that we'll talk about. We're going to look at transposable elements. And so these are elements that can move throughout the chromosome. So these are pieces of DNA that move. And this happens naturally. It was actually discovered by a woman scientist, Barbara McClintock. This we can actually cause to happen in the lab now, right? So we know how it happens naturally, and we can use it in the lab, in laboratory settings. So we'll look at both of that. We're going to look at gene transfer, and within gene transfer, we're going to look at three main types of this transfer, where DNA literally moves from one cell to another bacterial cell. Oops. Let's see. I already got it out. So this is another one that doesn't give me much of a warning, so I better get on it. Somebody put these in the outlet. It's not a storage device for markers. Okay, it's plugged in, it's happy, we're good. So we're going to look at DNA-mediated transformation, sometimes just referred to as transformation. Transduction, and those sound very similar, right? Transformation, transduction. So if you listen to my previous lectures and too, I give you guys some tips on how to, and I'll reiterate that. And then conjugation, and then the last one is regulation of gene expression. And now we're not going to go into too much detail, except I'm going to give you a couple of real-world examples right now so that you can understand the concept. So what do we mean by expression? Right, to show a feeling, right, to what's shown, right? So DNA, right, if a cell has DNA, what does it do with that DNA? Central dogma. DNA gets transcribed into messenger RNA, which is then translated by the ribosomes into proteins. So that protein is the expression, right? The gene is that segment of DNA, right? A segment of DNA that codes for a specific protein or gene product is referred to as a gene. So, in your body, in your cells, is all the DNA necessary to be any cell in your body. So red blood cells, when they're erythrocytes, before they lose their DNA in their nucleus, make a particular protein in very high concentrations. What do they make to be a red blood cell and do their job? Hemoglobin. They make buku amounts of hemoglobin. Liver cell. Does he know how to make hemoglobin? Is that encoded in his DNA? Yeah. Does he make it? Not really. Why not? He doesn't need it, right? His environment, the cells nearby say, hey, we don't do that, dude. We're liver cells. We got to do liver cell things, right? Like make bile and stuff, right? The red blood cells, on the other hand, where they're born and where they're programmed, that environment says you're going to be a red blood cell. You're going to make massive amounts of hemoglobin, right? It's somewhat environmental, you could say, right? Makes sense. why a red blood cell does what a red blood cell does, and liver cell does what it does. Although they, have all the information to be anything they want to be, right? They're told otherwise. That's a multi-celled organism, right? We need people or cells to be doing certain things to all coordinate to make these beautiful bodies we have. But a bacteria is a single-celled organism, right? That's it. One cell. So that cell, does it still have to deal with environmental issues? Absolutely, right? The environment actually really dictates all of the life of that poor bacterial cell. So imagine your cute little E. coli living in your intestines. That environment of your intestines is a particular environment for him. When you eat something that contains the sugar or drink something that contains the sugar lactose, normally what happens is you break it down using lactase, an enzyme that breaks apart the two sugars that make up lactose, which I think are maybe glucose and galactose, but don't quote me on it. You should know that though. But you break those two sugars apart and you absorb them in your small intestines and, they go throughout your body and feed your cells. Some people become what's referred to as lactose intolerant, right? They no longer produce this enzyme. So doesn't get digested in the small intestines. Instead, the lactose makes it to the large intestines where E. coli is. And I got news for you, E. coli, he'd love him some lactose, right? He loves that sugar, right? He has receptors in his membrane that tell him when lactose is present and allow him to take in lactose. And then he's going to make even more doors for lactose, right? He's going to make more proteins, receptors in his membrane that will bring that lactose in. And then he's also going to make all the enzymes necessary, so like lactase, to break down that lactose and utilize it as a food source. Does he always make those enzymes? No. When is he going to make those enzymes? When the sugar's available. Is lactose always available for E. coli? No, that would be a waste of energy and resources if he always made those enzymes to digest lactose. And if you're not lactose intolerant, is he ever going to see it? No. He just wasted all of his energy producing something he didn't need. So that's regulation of expression. He has the DNA. He has the ability to make those enzymes, but he's only going to make them when that sugar is present. And we use a similar system if you're taking the lab. We use a similar sugar digestion system that's been modified when we do the last experiment, well, the second to last experiment in lab, when we do the bacterial transformation, where we're going to actually put DNA into bacteria and cause it to express a protein called green fluorescent protein. But it's only going to do that in the presence of the sugar arabinose, because we're using that operating system that they use for that sugar. But instead of making the enzymes, we tricked him. He won't make the enzymes, he'll make green fluorescent protein. Right? We switched out the DNA. We switched out the genes. We didn't. The manufacturer did. Right? In the DNA that they give us. And the DNA we're going to use is in a circle. It's a plasmid, like we've talked about previously. But once you have information, you can use it. You don't always have to use it, right? You can choose sometimes when you're going to express it based on environmental factors like the fact that lactose is actually present. So the bad news with people who become lactose intolerant, what did I say about lactose and E. coli? He likes it. So if he has a food source he can utilize, what is he going to do? He's going to have some babies, right? They're going to grow exponentially, really fast. Now, does your E. coli usually grow that fast? No. But if you give it a whole bunch of lactose, man, is he going to grow. So then you have people that have intestinal issues because of this overgrowth of the E. coli that's naturally found in the intestines. So they experience things like constipation or diarrhea and gas, because it'll ferment the sugar and it produces gas. So once they figure it out, they usually stop eating things that contain lactose because it's really uncomfortable. Or you take the enzyme when you take the food so you can break it down and absorb it in the small intestines and not have it go to the large intestines. No buffet for the E. coli, right, anymore. Make sense? So one of the discussions that came up, and I think I looked it up in my last class, but I don't know if we talked about it. So they're like, well, some people say, well, we were never meant to drink cow's milk to begin with, right? And we know milk is a source of lactose. But lactose, that sugar, actually gets its name from the fact that it's lactated. It's put into milk by mammals that make milk for their babies. So in fact, women make lactose as well. So I mean, like, human milk contains lactose as well. We make that sugar for our babies too. So it's not all about like the fact that we're, quote unquote, stealing from cows. It is a natural sugar that all lactating mammals make. So why is it that, some of us stop making that enzyme? It might be because maybe we are at a certain point not supposed to be drinking milk anymore. That belief I do kind of believe in. Right? And that I just don't like milk, so. I like milk products, though. I'm glad I'm not lactose intolerant. I like ice cream. Funny story for y'all. At UConn University in Connecticut, where we were, they have their own dairy, and the students make ice cream. And they have their own dairy bar. It's called, I think it's called the Dairy Bar, or something like that. And it's set right, right on campus. So right before we left, one of the girls was like seriously addicted to the peach ice cream. So we went, a whole bunch of us, after the final send-off lunch, presentations lunch, we went for ice cream before going back to the dorm to pack up and get on the van to go to the airport. One of the people in the group, I get in my office yesterday, she sends around a message like, another link, here, this is cool. And then, who let the cows out? And there's a video. That afternoon, somehow some of the cows got out, and they were wandering the parking lot at the dairy bar. And I was like, thank God that didn't happen when we were there, because I don't know how long it took them to round up the cows, but I mean, we wouldn't have been able to get out of the parking lot to get to the dorm to get it out. And the guy from Spain, he's like, us in Spain, we've been known to let, cows and bulls run free, but I didn't do it. So, yeah, we had fun. And fun group. Definitely. I'm a tweeter now. They convinced me to get a Twitter account. Still learning, though. So, you can follow me if you want. I'm NolaSwee1. Nola_Swee1. And I'll be posting all cool, microbiology stuff. Mostly pictures. I thought Instagram would be a better place for me, but apparently Twitter and Instagram are competing. And so Twitter allows photos where they didn't used to. And videos, I think now. I'll probably be posting photos. Yeah, and retweeting, but not excessively. There's one person on the list I was like, tweet, 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 tweet. Do you ever shut up? So, yeah, okay. I'm learning. And then I also learned on Facebook during our social media thing that if any of you guys have had Kendra Anspa, she's moving back to Michigan. She resigned from Delgado. Yeah, I know. But in her defense, she did send me a personal email before she posted it on Facebook. I was a little pissed at first. To have found out my colleague, someone in my department was leaving, and I found out on Facebook. So, yeah. She's going back to Michigan. She'll be crying when it starts snowing. I won't visit her when it snows. I'll go in the summer. And she'll come here in the winter. So why are we talking about this? So I kind of alluded to this, and like I said, it definitely very relates to why I went for the workshop that I did. So we obviously want to be able to identify the different types of mutations, and we're going to go through those. But really the core theme that we want to keep thinking about as we talk about these mutations is their effects on the cells, right? So how does base substitution affect a cell? How does silent mutations, all these different mutations, and chemical mutagens, and radiation, and UV light, and X-rays. And she's got hers plugged in. That happened in our meetings too. Don't worry about it. One time one of the speakers got up and she's like, oh, I have theme music. It happens. So again, we're going to think about their potential effects on the cell. So we're going to talk about the DNA changing, but what effect does that have on the cell? Well, remember, why they can do things is encoded in their DNA, right? How they make enzymes is encoded in their DNA. So if that DNA changes, and therefore the proteins they're making change, can that be a good thing? It can be. Can it be a bad thing? It can be. Can it have no effect? Yes. So that's the main gambit, right? It could be really good, like winning the lottery good, right? They could become resistant to an antibiotic. That's really good for a bacteria. Granted, from our perspective, that's really bad. But for them, that's really good. They're going to survive, right? It could have no effect, really no change. It could be really bad for them, right? This change could result in them not being able to do something necessary for life and they could die. Those are the extremes. And it really varies depending on what changes and how it changes. So we're going to look into the specifics. Like I said, we're going to define these things. What's a base substitution? Within that, what's a silent mutation? What's a missense? All of these things. And before we can even really talk about that, we have to look at the fact that we're talking about prokaryotes, right? And they're haploid. And because of that, their mutations usually lead to phenotypic changes. Explain why. So let's first define some terms that you may not recognize, right, from previous biology. Anyone know what haploid means? Nope. 1. We are 2, right? We are what's called, eukaryotes are what's called diploid, right? We have 2 what? Yes, sets of DNA. You could say 2 sets of DNA or 2 sets of chromosomes, right? Why do we have 2? 1 from mom and 1 from dad. And that's the good news for us, because if mom gives you, say, a defective copy of hemoglobin, maybe your dad saves your ass and gives you the good copy. But that's why you have sickle cell disease, right? It's a defective copy of hemoglobin. It doesn't aggregate correctly in the cells, and it deforms the cells into that sickle shape. Well, there are some people which are called heterozygous. They have 1 good copy and 1 bad copy. But you make so much of that protein, you really need 2 good copies. So they don't have full-blown sickle cell anemia, but they do end up with some sickle-shaped cells. But this ends up being environmentally good where and what part of the world? Yeah, it makes them relatively resistant to malaria, which is found in Africa. So that's why people in the African population, unfortunately, a lot of them also have sickle cell anemia, because people who are heterozygous, just a little bit of sickle cell, so they don't have sickle cell anemia full-blown, but they are more common in the population because they don't die from malaria. So then they have kids, but then when you draw that Punnett square, right? Remember those? The reason why we draw them is, again, you have 2 copies. Everybody's got 2 copies, 1 from mom and 1 from dad. So I'm going to do that S for the good hemoglobin, right? And little s for the bad hemoglobin. And do cursive because you can tell the difference. So if both parents are heterozygous, they're going to give to their children either the good gene or the bad gene. And so when you fill in your square, I can't even do cursive X's. I got to think and not talk, because otherwise I screw up the chart. Big S, big S. Big S, little s, little s, little s, little s. So, about 1/4 of the children have a chance of getting both normal genes being 100% normal. But in Africa, that's not good, is it? Because you're susceptible to malaria then. Because you don't have those sickle cells that prevent the plasmodium from infecting those cells. Half the children have the potential of being heterozygous like the parents, which means that some of their cells sickle cell, but not enough that they end up with anemia. They have enough normal cells that they're okay. And the added benefit is they're resistant to malaria. 1/4 of the children, unfortunately, will have full-blown sickle cell anemia. All their red blood cells sickle cell, right? So they form that sickle shape and they clump up in arteries and they really don't do their job like they're supposed to. So that's why in the African American population you see a lot of this defect has been perpetuated because this form, heterozygous, is beneficial. But most genetic diseases, again, you've got to be homozygous for the recessive, or what we refer to as defective gene. Recessive, right? The good news is for most diseases, you've got to have both copies bad to have the disease. If you're heterozygous, usually you're okay, but it really depends on how necessary that gene is. Like my husband is what they would sometimes refer to as a carrier, right? Like this would be like a carrier where they have one bad gene but one good gene. He has hemochromatosis. So he has one, I kid you not, one nucleotide screwed up in his DNA, right? And so his body keeps too much iron in his blood. That's actually can be bad. Like anything in excess, right, is bad. And only one of his genes is screwed up. And he got it from his mom. So he's got to go every couple of months and they just take a pint of blood and throw it away to get rid of the excess iron in his blood. He has to limit the iron in his diet. He can't take vitamins that contain iron, things like that. Because it will build up in your liver and toxify your liver and other organs over time. Can't donate blood even though he's like O negative. They would love to have his blood, but they can't have it. Sucks. Right. We also chose not to have children because, I mean, we've got a whole bunch of this crap. And we pass it on to our kids. I'm already like would have been deleted, you know. If natural selection actually affected the human population like it does other populations. Does this make sense to you guys though? So we have two. Do bacteria have moms and dads? No, huh? Pretty much just mom or dad, depending on how you want to look at it, right? One parent, let's put it that way, right? They have one parent. They are an exact copy unless there was a mistake made in that copy, and they're a clone, they're an exact copy of their parent. And the problem with that is how many copies of, like, say, how to make, they don't make it, but hemoglobin do they have? One. Typically only one. So if that is a defective copy that their parent gave them, are they going to be able to survive? Depends. Depends, right? Depends on what it is that's defective, right? I mean, my husband's still alive. He's got defective gene for the whole iron thing, right? But he's still alive because, you know, we can treat that. If you're a bacteria and say, now you can't make lactose anymore. If you're E. coli and your gene for making lactase to digest lactose is defective, so you no longer can digest lactose, are you going to die? No. Are you going to be able to digest lactose? No. So when the lactose party comes, you can't participate, right? But, you know, it's not the end of the world. Does that make sense? So you see how this can vary? It really depends on, like, did you really need that gene? In the case of lactose digestion for E. coli, not necessary for life. It'd be beneficial, right, to be able to party with the rest of your buds when lactose comes online. But you're not going to die if you don't have it. Make sense? So the problem, too, is like, you know, one copy. Again, if it's defective, that could be really bad. But because of that, they change faster than us, don't they? Because if it changes and it's good, can you imagine, like, they've got a better lactase enzyme. So when the lactose comes, man, they can digest it 10 times faster than the E. coli buddy next to them. So they're going to predominate the population, right? Because they're better than the normal lactose E. coli digester. Make sense? Yes, maybe so? Okay. All right. So what are mutations? They're stable, heritable changes in the DNA sequence. So the change happened, it didn't get fixed, it got passed on, it's heritable. It's given to the next generation. In prokaryotes, usually produce phenotypic changes. So we already said they're haploid, right? So they have one copy. So that one copy is used to make the messenger RNA and make that protein, say lactase. What is a phenotype? It's the physical expression of the gene, right? So in most of the time we talk about like characteristics like blonde hair and blue eye stuff. That's actually genetically, like at the DNA level, multiple segments of genes that actually determine these things. But when we talk about a single protein, like say the enzyme lactase, we really are talking about a single gene. And so that enzyme is the phenotype. The fact that you can make it, right? You would say that this E. coli is lactase positive. If somebody mutates and no longer can make lactase, you would say that that strain is lactase negative. Make sense? Right? And again, we're referring to their phenotype. What can they actually make from their DNA? Or not make in the case of not being able to make an effective lactase. They may make it, but it just doesn't work too. Or they might not make it at all. Depends on how screwed up the gene is, the DNA is. These changes can occur spontaneously, or they can be induced by chemical mutations or radiation. So we know, right, that chemicals can change DNA, they can mutate DNA, and the greatest fear is that they mutate it to the level that the cells become what? What do we fear the most? Cancer, right? The cells behaving badly is what I call cancer cells. They don't do what they're supposed to do anymore, and worst case scenario, they do shit they're not supposed to do. Right? That's why they're so bad. So we purposely test chemicals to see if they mutate, if they change DNA. If they change DNA, would you want to take that chemical or that drug or use it? No, because there's the potential it could change it to such a degree that it becomes a cancerous cell. And so we'll talk about that. Radiation all the time mutates your DNA. It's kind of terrifying, right? How many of you go to those tanning beds? Good for you. Bad idea. It's UV radiation, y'all. It mutates your DNA. Yeah, it makes your skin darker, but it mutates your DNA. So the more and more you do it, what's the chances that you're going to generate cancer cells? It's higher. Yeah, those people that own tanning bed things probably don't like me very much. You can go, if you've got to be dark, you can go get the spray tan, right? But then again, how long have we been doing that, and what are those chemicals doing? Another thing to think about. White is beautiful. Lighter pigment is beautiful, right? It's also very healthy, hence why I went and bought sunscreen, right? I told you guys about Dean Gruber, right? If you've met our Dean, look at his nose. Cancer cut off 2 or 3 times, right? My dad, his nose cut, like half his nose gone due to standing outside coaching baseball and not wearing sunscreen for years on end. Not a good idea. Every day you go out there, you're exposed to UV light. It's always mutating your DNA. The good news is why we're not all dying of cancer tomorrow, right? And of course this happened to them in their 40s and 50s, right, because of long-term overtime exposure. You have repair mechanisms, right, that do repair your DNA. But if you overexpose it, you just keep doing it, keep doing it, keep doing it, eventually, right, they're just not going to be able to keep up with the bombardment. Radiation, what else do you think of as far as radiation? X-rays, right? Are you going to go every day and go get x-rayed? Not a good idea, right? And that's why, too, if you don't have to, right, what are they going to protect? When you go for x-rays, when you go to the dentist, what do? They put this big old lead vest on you. Why? What are they trying to protect? Your ovaries or your testes, your sex cells. Because if those mutate, you end up with mutated children. I don't know about any of you, but I don't want a mutated child. It's also why when you're pregnant, they're not going to x-ray you unless they absolutely have to. Because again, you're going to x-ray that baby too. Ultrasound is different, right? They're not flinging electrons at you with ultrasound. It's sound waves that are bouncing back and being visualized. Safer. Make sense? Okay. So, but yeah, they're throwing electrons at you. They're stripping and potentially damaging your DNA. For the most part, if you do that once every couple of years, you're going to survive it. You don't want to do it if you don't have to. That's why when the dentist's like, we're going to do x-rays, I'm like, really? Again? Is it really necessary? Right? And that's why they leave the room too, right? They don't want to get irradiated. Okay, so these mutations happen spontaneously. They occur infrequently and randomly. And the range can vary and the rate at which it happens extensively. The reason why it's not very high, the rate, per se, is because, again, most cells have repair mechanisms. Because, again, DNA is precious. That's your information on how to do what you're going to do, when you're going to do it, how you're going to do it. And you don't want that information damaged. Right? You want it protected as best you can. It also really depends on the cell. I mean, think about it. If that cell multiplies every 20 minutes as opposed to one that only multiplies once a day, what's the chances of the one that multiplies every 20 minutes? Like, his probability goes up, right? Because he's copying so often. Where the guy who only does it once a day, but then again, what's the rate? How much DNA? I mean, there's so many variables, it's ridiculous. But for some cells, we actually, smart people have calculated out their mutation rate. How often do they mutate? So these animations can be good. And did you guys do your, I spied on you a little bit, but my brain is, oh no, that was my other class I spied on in Connect. Did you guys do the bonus? Did you guys watch these animations yet? No. All right. We'll do the fast version. I'm going to fast forward and do my take on it. I'm not going to play it. In presentation mode, which I'm in, you can hit the play button and we'll actually play. But I'm not going to do that. I'm going to just move it so the pictures move. All right. I'll do my own audio. So here you have just the nitrogenous bases of the nucleotide, right? And we know, right, in DNA, they hydrogen bond with each other, the two parallel complementary strands, right? So you guys remember for DNA, it's A's with T's and G's with C's, right? So what do we got here? It's probably an A and a T because they only have 2 hydrogen bonds. G's and C's have 3 bonds. Not anything you need to know for this class, like if you go into genetics maybe. So the other thing, see, I guessed right. Adenine and thymine, A and T, have the 2 hydrogen bonds. And then when you look at adenine, this one falls into the class known as purines. They have 2 rings in their structure, where pyrimidines only have 1 ring. And there's a little saying that was taught to me a while back on how to remember which nucleotides are purines, is angels are pure, and they have 2 wings, right? So A and G is in angels, right? So adenine and guanine are purines, and they have 2 rings, just like you think of the 2 wings that angels have. You don't have to believe in angels. They could be fictional characters too. Does that make sense to you guys? Again, not anything I'm going to ask you in this class, but like if you go on to genetics and stuff like that, I'll kind of help you out. So, and then the pyrimidines, I always kind of, some people say, think of a pyramid, think about just one structure, one point. So one ring for pyrimidines. So purines and pyrimidines always bind with each other. So if you think about the structure of DNA, that step, if you think about the width of that step, that where the nitrogenous bases are, I always imagine DNA as a spiral staircase, right? Can you imagine a spiral staircase? So each one of the steps are the same width because you always have purines and pyrimidines binding with each other, so you always have 3 rings across. So you can imagine the step is always 3 feet across. It always has the same width. And this is partly how Watson and Crick figured out the structure of DNA. Because they found that you always had the same number of A's as you did T's, and G's and C's. And then, you know, you're looking at and then of course I look at Rosalind Franklin's work as well, and right, they're able to help figure out this structure. This structure is very important to the functionality of our cells. Because what happens is you've got those nitrogenous bases, but what are they hooked to? What does DNA stand for? Deoxyribose nucleic acid. What's a ribose? It's sugar. And in the case of DNA, that sugar is missing an oxygen that normal ribose would have. So you have the nitrogenous base, whether it's adenine, guanine, right, thymine, this portion, attached to the sugar, and then to what? P? No. Phosphate. Right? So it's the chemical name for the P. No. Periodic table. C is carbon, N is nitrogen, P is phosphorus. Thank you. Phosphorus with oxygen is a phosphate, right? With 4 oxygens, right? So nitrogenous base, sugar, phosphate. How are the nitrogenous bases hooked to each other? Phosphate binds with sugar, which binds with the next phosphate, which binds with the next sugar, right? So a lot of times that's referred to as the sugar-phosphate backbone. So now go back to your spiral staircase. What part of the spiral staircase is the sugar-phosphate backbone? The railing, right? The structural support that holds the stairs. Can you see that in your mind? So what do little kids do when they see a railing? They slide down it. Guess what your enzymes do? They hook on to that sugar-phosphate railing, that backbone, and they literally hook onto it. And they literally slide down it. And as they're sliding, they're reading that code that's on the steps as they go down. So, can you imagine if you hired a carpenter, brand new house? Yep, I'm going to make a spiral staircase. I want the first 3 steps to be 2 feet wide, and the next 8 steps to be 4 feet wide, and then I want them to be 2 feet wide for another 12 steps. What's that carpenter going to say to you? You a crazy lady. Right? It's not possible, is it? You couldn't do that. It wouldn't be structurally sound. You physically would not, it would be very difficult, let me put it that way, to do that. And think about that, it would be like this, right? Going in and out in a spiral. Forget about it, right? So in your cells, if they make these switches that we're going to talk about, and say their step becomes only 2 feet, or then it becomes 4 feet wide, because they put the wrong nitrogenous bases there and it's not 3 feet anymore, it's not 3 rings, 3 rings, 3 rings, then it's going to distort the structure of the DNA, isn't it? So are they going to be able to slide down? No, they'll fall off. You know, and so what do we do nowadays at escalators to keep kids from sliding down? We purposely put bumps and barriers, right? So they can't slide down. Not easily anyways. Right? But when it comes to DNA, we want that whole sliding. We want them hooking on, sliding, and reading that code. So any distortions that lead to actual distortion in the DNA, usually our enzymes are going to detect that and fix that, right? Because that's going to screw up the whole system. Still with me? All right, so let's look at this at the molecular level. So here, these guys are normal binding, 3 rings across. So let's say, you know, this hydrogen hops over here. So this adenine, who's supposed to bind with who? You can say the letter thymine, T. Now can bind with a different pyrimidine. So this is okay because we're still talking about 3 rings across, right? For our step. So our step is structurally sound, but is he supposed to bind with cytosine? No, it's supposed to be thymine. So is that going to mess up our code? Yeah, if we don't fix it. So these particular changes are given specific terms. We're not going to go that far. I don't want you guys memorizing these terms, but I want you to kind of understand this concept, right, of these switches and how they affect. Does that make sense to you guys? So this is just changing out just one, and we're going to talk about how this can actually lead to a phenotypic change, or even worse. So here, now the next one, we've got thymine. It's switching up and binding with guanine. Again, still 3 rings across, but again, not who he's supposed to be binding with. It's supposed to be adenine here. So here, right, adenine binding with cytosine instead of thymine, right? This gets passed on, and when we look at the code, that mistake of that C and then now the G getting put there is what we call a mutant. It changed, right? And it got passed on. And notice that the codon, these 3 nucleotides in RNA, is then would normally be the wild type is what's normally in the environment, the most predominant type in the environment, hence wild. The one that's different is referred to as the mutant because it's mutated, it's changed. Notice that this codon now, that group of 3 nucleotides in the messenger RNA codes for a different amino acid. So it's like, oh, okay. It's only one amino acid. What's the big deal? Is it a big deal? It can be. Again, this falls into that area of like, how important was that one amino acid? Does that mean that that was in the maybe the active site of the enzyme, and now the enzyme doesn't bind to the substrate and it doesn't work anymore, right? You now have defective lactase because of that one amino acid. That could happen. Or now it's super lactase. It's like super fast. It binds with the lactose even faster, and like they're like Superman now. Or the protein won't even fold anymore correctly because that one amino acid was really important to its folding, and it can't make its active site anymore, and now it's basically no good. Those are 3 real distinct possibilities that could happen from just changing one amino acid. One nucleotide, excuse me, that changed one amino acid. So as I said, they talk about, you know, tautomerization resulting in transition, where you sit what we saw, which was one purine for one purine or one pyrimidine for another. But sometimes you'll get these transversions where you're actually switching a pyrimidine for a purine. And that's really bad because that's going to distort the DNA. So those are usually don't make it through. But whenever we're changing just a single base, that's known as a base substitution, right? We're switching one for another. So spontaneous mutations occur without exposure to external elements, right? They're just copying the DNA and it screws up. And it doesn't realize it made a mistake and does not fix it, right? So that you can end up with these base substitutions, or you can have insertions or deletions, which is one of the things we're going to look at in a moment. And you may even get mutations because DNA was lysed, like that sugar phosphate backbone was broken and the pieces get jumbled up, right? It's like a jigsaw puzzle, right? without a picture. Have you ever tried to do a jigsaw puzzle without a picture? It's not very easy. Right? So here, again, just pictorially, right? Not binding with who they're supposed to bind with. So that's from the animation. And then here, showing again where the change happened and it got passed on, and now you have this mutant. So within substitutions, base substitutions, we're going to look at just a single point mutation, a single base being switched out for another. We're also going to look at adding or removing nucleotides, the effect that has, and as I said, transposable elements. So within base substitutions, as we said, there are possible outcomes that can happen. Normally, the wild type, right, the DNA should code for this. This should be your codon in the messenger RNA, and this should be the amino acid that the ribosome puts there. But if we say change this one, right, this TA combination for a CG, we get this codon. And if you look up this codon, although it's different from this one, right, this one's a C, this one's a U in this third position, if you look at the chart, it still codes for the same amino acid. So we refer to this as a silent mutation. Why do we say it's silent? Doesn't change what? Doesn't change the amino acid, doesn't change the phenotype. But is it a mutation? Yeah, because what changed? The base where? In the DNA, right? A change in the DNA that's not repaired or fixed or changed to the correct, that's passed on to the next generation, is a mutation. But in this case, it's silent. So this would have no effect on the cell, right? It's still going to make the exact same protein it used to make. It's going to function exactly the same as it used to. But what if that mutation happens here, say, and now we get a different codon, but now this codes for a different amino acid? First of all, this is called a missense mutation. Again, it's a mutation, right? It's a change in the DNA. Missense, remember, codons that code for amino acids are called sense codons. In this case, is it the correct amino acid? No. So we refer to this as a missense. So I don't know if it was made on purpose, but it definitely helps me remembering that basically it's a mistake, right? Miss means you messed up or you missed, right? You got it wrong. What effect will this have on the cell? Depends. Could this be bad? Could this be like really bad, lethal? Yep. Could it be good? Yeah. It could be super lactase, right? Could it have no effect? Yeah. Could be still lactase, still functions the same, no problem. Could it stop working? Sure. Would that necessarily mean death of the cell in this case if we're talking about lactase for E. coli? No, they can live without lactose digestion, right? It's not necessary for life. So you guys see, right, all the possibilities? Now this next one, the codon gets changed into a stop codon. And what we call stop codons is nonsense codons. Right? So they don't code for anything, right? They tell the ribosome to do what? Stop. Do you think this could be good? I don't know. If you stop in New Orleans at a yellow light, what happens? If you're not paying attention to the person behind you and they're not paying attention, they rear-end you, right? Inevitably. Right? I see a yellow light, I look in my rearview mirror first before I stop. All the time. Especially if it's raining out. So if you stop, potentially before you're supposed to, right? That's what would happen here. You're not going to stop after, right? Because the natural stop is before that, right? So inevitably, if we've got a stop codon before the normal stop codon, is that going to be good? very, 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 I keep going, rarely is a stop codon mutation good. Because you're going to not make the whole protein, right? Well, if you didn't need to make the whole protein, why would you ever make the whole protein? Right? Why waste amino acids if you don't need the whole thing? You need the whole thing to make a functional protein. So stopping before you're supposed to usually ends up in not making a functional protein anymore. Again, is that going to kill a cell? It could, if they really need that protein, right? And now they don't make it right. It doesn't work. Could it have no effect? Or maybe put them at a slight disadvantage, right? Especially if we're taking lactase as our example, right? Now they don't make functional lactase anymore. They're not going to die, but they don't get to go to the lactose party. Make sense? Okay, ready for a break? You guys look ready for a break. Yeah. 15 minutes, so